So Chris joined my lab a little bit over a year ago in December, December 15th. So prior to being a master's student, Chris was a high school teacher for 4 years. And so when Chris had approached me about working in the lab, I sort of thought about, well, if you're a high school teacher, you probably have a lot of patience, perseverance, but you also have to really have a positive attitude, right? And so he's really made great progress. And you know, I think he's the type of student that all PIs wish to have in their lives. Very positive attitude. I think it was very infectious among other students in the lab, especially among, with me. Every meeting he would come in and, experimented at work, it didn't matter, he was going to forge ahead and figure it out. And that proved true about midway through his thesis, where we were trying to develop some technologies which I discussed, but then he came to me with an idea of a new technology that had just been published. And he wanted to take this risk of sort of adapting this new technology to the lab, right? And I had my doubts about how easy this would be, but he proved me wrong about how he could just get this ball rolling very quickly. And so, I think it's, I'm sad to see him go, but it's, I still present, he's done a lot of nice work, and he's trained another master's student who's going to, reap the benefits of sort of the hard work and effort he's put into it. So, without further ado, so Chris.
Can I play some rules?
Yeah.
I know you like him, but so my name is Jim Liu. I'm the professor in the physics department, but I'm the director of this particular program called Professional Science Master's Degree. Okay, Chris joined us actually I view as a senior experienced person because he had taught students for a few years. So throughout the program, like you said, he always, doing things to bring other people on board. So I really appreciate that. So he was in my class for years. We had a lot of conversations, a lot of dialogue, and today this is the final one. So I'm also very glad to look at that professionalism, right? You and I, we didn't do that. But that's fine, because in my class, we talk about professionalism all the time, right? Not just like professors, you don't have to dress up, but when you go out in the community, in the world, you need to think about which, situation you need to be professional. This is appropriate. Now, in our program, we also have a rule. We negotiate on how long they give presentations. So he said 15 minutes, I said 45. So now we're going to go for 45 minutes for his presentation. This is very strict. Okay. The other one, the rule is that he will.
Ready? Thank you guys all so much for coming. In case you're unaware, I am Christopher Potts, and I want to tell you about my project, which was generating these isogenic ApoE variants in order to look at Alzheimer's disease risk and progression. So immediately, Alzheimer's disease is a neurodegenerative disorder. It's the most common form, which generally leads to this cognitive decline. Now, one of the issues we have with Alzheimer's disease is that while these hallmarks are present, and amyloid beta plaques and neurofibrillary tangles have been shown to be neurotoxic, the actual mechanism through which Alzheimer's disease manifests is still unclear. Most of the indications we have for how it works come with effects which have been linked and mechanisms which have been linked to the progression of Alzheimer's disease. Everything from efflux of cholesterol, mitochondrial dysfunction, neuroinflammation, tau pathology, and even neurotoxicity, which have then been linked to Alzheimer's disease. However, these linkages have not proven consistent throughout all populations. Pluripotent stem cells, or iPSCs. Now, these are great because they are human, and we can manipulate them quite easily. We can control a lot of things about them, their environment, which genes they express. We can alter them, and we can control the difference between these cells. Because our iPSCs are grown up as clonal lines, they're genetically identical to one another. And what this means is if we alter the ApoE gene to look at these differences, we will know that any differences we see in expression or morphology are due solely to this ApoE isoform change. Now, the one big drawback is that these are grown in a dish. They don't have the complex microenvironment offered by primary tissue or mouse models, which some would argue has more of an effect than others. But they're still the most effective and efficient way to look at RNA, or SG, short guide RNA. Now, this short guide RNA will target a complementary section of the DNA, making cloning a lot easier because we're relying on Watson-Crick base pairing in order to target our gene of interest rather than these complementary protein sequences. And this has the added advantage of cleaving both strands rather than a single strand requiring two reverse complementary domains to be generated. And so we began by using CRISPR to try to modify the ApoE gene. So to look at this non-homologous end joining, we use this assay called a T7 assay. I just want to explain to you how that works real fast. So we have our heterogeneous population, most of these cells being wild type, with some being genomically edited. What we do is we amplify our gene of interest through PCR, and some of these PCR products are going to have this indel from our non-homologous end joining event. We denature these pieces of DNA and then randomly reanneal them. While most will reanneal to themselves, some will have a length mismatch, which will result in a little bubble where that indel is. We can then add a T7 enzyme which cleaves the DNA at that length mismatch bubble, giving us most of our pieces of PCR. So what we did is we designed primers which flanked our GFP target region and would cause PCR amplification opposite the direction of our insert. Now, one of our primers has this little tail on it, which contains our new target sequence. So after amplifying this through PCR, ligating it back together, and doing a DPNI digest, which removes the initial plasmid, we're able to generate our new ApoE-targeted guide RNAs. Here's our original sequence. Here's the first sequence. Now, what you'll notice here, this is the sequence for changing the cysteine or the arginine mutations that we want. So then we look to transfect into iPSCs. Again, when we tried this the first time, they all died. And so we proceeded, and this was, I should also mention, with the 112 conversion marker, which goes from E4 to E3. So because they all died when we added all three of these, we went back to just adding the GFP marker and trying to optimize this transfection. And we tried a lot of different variables here, our cell density, our number of pulses, and what we saw was really, really exciting. When we took these populations after 3 days, 3 days post-transfection, we sorted them into GFP negative and GFP positive populations, and then sequenced them, we see in the GFP negative, there is no appreciable base editing here, converting the cytosine, this blue peak, to a red peak. But in our GFP positive cell lines, we see a huge spike in our thymine conversion, especially when compared to our unsorted cells. Now, this is Sanger sequencing and should not be considered quantitative at all, but this does indicate to us that sorting these cells into GFP positive and GFP negative populations can increase our ability to isolate. We're also trying to generate a guide RNA GFP hybrid vector, where we're taking the guide RNA cassette and putting it onto the GFP marker vector, so that way we only need to transfect with 2 plasmids instead of all 3, hopefully, again, increasing our cell survival and transfection efficiencies. And then we want to validate that this works in iPS cells. Long term, though, we're going to look at isolating these isogenic clonal lines. And once we have these clonal lines, then we can look at neuronal differentiation and doing expression analysis and comparison on them. Thank you all.
okay, including group members, because others, they may not know what they ask, but the group members, you know. So, any general question?
So once you have isogenic cell lines, for how long?
That's a good question. Theoretically, indefinitely. One of the hallmarks of being a stem cell is that these cells are effectively immortal. And when passaging these cell lines, occasionally you will get random mutation events, but the occurrence of that is so low that it's not difficult to maintain these isogenic populations. We also, anytime we have a new cell line, we grow up a bunch and freeze back stocks of them. So even if one line becomes mutant and different, we can go back to our frozen stocks and use those. So effectively forever.
Any other questions? Okay, I can ask a general one. Later on, we'll ask a different one. So humanity GFP positive or GFP negative, they have so much difference in transfection.
Yes.
So do you think that GFP affecting anything?
I don't think the GFP protein has an effect on them because the GFP expression doesn't start until about a day after.
Multiple ones.
Yes.
You have multiple targets.
Not multiple targets, just since.
Well, one is the efficiency, right? One is the
Yes.
okay, I take more. The other one, you imply you have multiple targets or this high efficiency.
When we're looking at transfection efficiency, we're essentially only asking how much of this plasmid got into these cells. This guide RNA plasmid we co-transfect them with only has the one ApoE-C158R target. So it's only going to target that one. Even if it gets multiple of that, it's still just targeting that one spot.
Yeah, I get concerned if I have a residual targeting, you know.
Yeah.
Then you have residuals, then you will have a problem later on.
Absolutely.
I have one just general. So you're generating the isogenic lines. You've done kind of one so far, right? Does it already have any familial Alzheimer's mutations, or is it one of the non-demented?
These are, so the cells that we're doing these in are all non-demented or sporadic Alzheimer's patients. That said, there is a lot of research that shows that ApoE compounds the familial mutations. So people with familial mutations that also have an E4 allele are more likely to develop Alzheimer's earlier in life, and say a familial mutation in conjunction with an E2 allele will actually delay the onset of that familial Alzheimer's.
Maybe a technical question about the protocols. So after you do the transfection,
Cas9, in part of the paper, they describe it as a catalytically active Cas9, and they reactivated it. Why do you think reactivation of it is important? And what exactly does that, what difference do you think it makes?
So the key part of that reactivation is that it's the editing. And so wherever our cytosine has been converted to this uracil, this reactivation cleaves the opposite strand. And what they've essentially theorized at is that this induces the cell to repair the opposite strand. And so rather than waiting for replication, which usually would result in this repair, the cell is actively repairing the opposite strand to be complementary to our uracil.
So to increase the rate of base excision repair and not have like proofreading with DNA replication going on.
Yes, exactly.